Good morning, students. Today we are going to discuss about transcription mechanism in eukaryotic organisms. So as you know, transcription can be defined as synthesis of RNA molecule from single-stranded DNA template in the presence of enzyme RNA polymerase. In eukaryotic organisms, three different types of RNA polymerases are there, which are responsible for the synthesis of different types of RNA molecules. RNA polymerase 1, which is involved into the synthesis of pre-rRNA, which is processed into 28S, 5.8S and 18S rRNAs. Second is RNA polymerase 3, that is involved into the synthesis of transfer RNA molecule, small and stable RNAs. And RNA polymerase 2, that is the main enzyme responsible for the synthesis of messenger RNA and for small nuclear RNAs which are involved into the RNA splicing process. So today what we discuss about the mechanism of transcription, that we will discuss with reference to RNA polymerase 2. Now, as you know, direction of synthesis of RNA, it is carried out in 5' to 3' direction. The DNA template strand is read in 3' to 5' by RNA polymerase and new RNA strand is synthesized in 5' to 3' direction. There are regulatory sequences at 5' region of the coding strand as well as into the 3' end of the coding strand. They are called 5' untranslated region, and the sequence found following 3' of the coding sequence, it is called 3' untranslated region. And this strand plays a significant role into the mechanism of transcription. The mechanism is divided into three phases: initiation, elongation and termination. And today we are going to discuss about initiation of transcription in eukaryotic organisms. Before we start initiation, we should know about promoter, because promoter is the sequence which is recognized by enzyme and it is the start site for the process of transcription. In case of eukaryotic organisms, promoter recognition is carried out by accessory proteins that binds to promoter and recruit RNA polymerase to the promoter. There are two groups of such protein molecules that play significant role into the promoter recognition and to initiate the transcription process. One group of protein, they are known as a general transcription factors. These general transcriptional factors recruit RNA polymerase at the promoter, and along with RNA polymerase, they form basal transcription apparatus which is assembled near the start site and that initiate the minimum level of transcription. The other group of proteins, they are known as transcription activator proteins. So keep in mind, two type of factors are involved. Number one, general transcription factors, and second are transcription activator proteins. Now looking to the structure of promoter, there are two types of promoter for eukaryotic organism. One is a core promoter, second is regulatory promoter. The core promoter is located immediate upstream to the gene. It is a start site and it is a site for the binding of basal transcription apparatus. This core promoter consisting of consensus sequences, and the most common consensus sequence into the core promoter is Tata box. This Tata box is located -25 to -30 base pair upstream to the start site. There is another consensus sequence which is located at -35 region. These are the two consensus sequences that are recognized by transcriptional factors, and basal transcription apparatus formation is carried out. The other promoter, it is known as regulatory promoter. This regulatory promoter is located immediate upstream to the core promoter. It consisting of a variety of consensus sequences. transcriptional activator protein binds to this consensus sequences into the regulatory promoter. These transcription activator proteins also bind to enhancers. We will see where the enhancers are located. So keep in mind, as we discussed two types of proteins, general transcriptional factors and transcription activator proteins. General transcriptional factors bind to core promoter, transcriptional activator proteins bind to regulatory promoter. Enhancer is located so far upstream to the promoter site. The DNA between the promoter and enhancer loops and transcriptional activator protein binds to enhancer, interact with the basal transcriptional apparatus and promote high level of transcription. We will see in picture how the process is carried out. So concentrate, this is the structure of promoter. This is the transcriptional start site. So keep in mind, students, that the core promoter encompasses few sequences into the promoter region and few sequences into the RNA coding sequences. This core promoter, upstream to the core promoter, there is presence of regulatory promoter. At -25 region of the core promoter, there is a Tata box. This sequence plays a significant role to initiate the process of transcription. There is another consensus sequence located at -35 region. And this is the regulatory promoter. 3 different sequences has been mentioned over here into the regulatory promoter, consisting of variety of consensus sequences. And this is the site where transcriptional activator proteins bind and promote high level of transcription. Now enhancers. Enhancers are located several kilobase pair away from the start site. They are usually 100 to 200 base pair long sequence consisting of multiple control elements. You can see over here, this is the promoter. At far distance there is enhancer. The DNA between the promoter and enhancer loop out, so enhancer come into the close proximity to the promoter and activate high level of transcription. And you can see over here these activators, they interact with enhancer as well as regulatory promoter. Now we discuss about the mechanism of initiation of transcription in eukaryotic organisms. We discussed about the structure of promoter, we discussed about the enhancer, factors involved, 2 categories of proteins, general transcriptional factors and transcriptional activator proteins. These all form basal transcription apparatus at the transcription, that is a core promoter near to start site. And this process, that is a formation of basal transcriptional apparatus, it is considered as initiation of transcription. Initiation is carried out by the assembly of RNA polymerase II and series of transcription factors. RNA polymerase II is associated with 6 types of the general transcriptional factors. They are designated as TF2A, TF2B, TF2D, TF2E, TF2F and TF2H, where TF stands for transcriptional factor, 2 stands for the factors involved into the role for the RNA polymerase II, and the last letter A, B, D, E, F and H designate individual factor. The 6 general transcriptional factors play a significant role to initiate the process of transcription. Among these 6 the first one is TF2D. This is very important. This TF2D consisting of TBP, one subunit. It is a multimeric protein consisting of 9 different subunits, and one subunit of this is TBP, which is called Tata binding protein, and other TAFS means TBP associated factors. As we have seen into the structure of core promoter, there was a Tata sequence that was located at -25 to -35 upstream to the transcription start site. The role of this TATA binding protein is the recognition of TATA sequence into the core promoter. And other these TAFS factors assist TBP in this process. In human cells, TAFS are formed by 12 subunits, and one of them is TAFS 250, having a molecular weight of 250 kilodalton. This factor has histone acetyl transferase activity, which is responsible or involved into the relieve the binding between DNA and the histone into the nucleosome. So as you know that in eukaryotic gene, DNA is bounded with the histone protein to form nucleosome structure. When transcription is carried out, this nucleosome disassembly is required, and this is carried out by TAFS. General transcriptional factor along with RNA polymerase, when they assemble at the core promoter, this process is known as formation of pre-initiation complex, which is very important to initiate the process of transcription. You can see over here variety of 6 different factors are there, as we had discussed: TF2A, TF2B, TF2D, TF2E, F and H. These all factors combine with core RNA polymerase 2 at the core promoter and form pre-initiation complex. Among all these factors, the first factor play a role, that is the TF2D, which contain TBP, that is a TATA binding protein, and this is playing a significant role to recognize the core promoter and initiate the initiation process. You can see over here, first of all, TATA binding protein of TF2D binds to the TATA box. Then TF2A and TF2B binds to TF2D. Afterwards, transcriptional factor F form a complex with RNA polymerase, and it bring RNA polymerase at the core promoter, that in turn binds with the TF2B. Transcriptional factor 2 F and E open the double-stranded DNA, and transcription factor 2H complete the formation of pre-initiation complex. So all these 6 general transcriptional factors sequentially attach at the core promoter. It bring RNA polymerase to bind at the core promoter and form pre-initiation complex. Now, if we see the role of all the 6 factors, TF2A consisting of 2 subunits, this has a major role to stabilize TATA binding protein and TF2D binding, and it blocks the inhibitory effect of TAF1 and the other protein molecule. The second is TF2B. This has a function to stabilize TF2D promoter binding. It contributes into the transcriptional start site selection, and it helps the RNA polymerase 2 along with TF2F to bind at the core promoter. Next one is TF2D. This is the major element that bind to TATA element at the core promoter. It provides a platform for the binding of TF2B and other TAFS and the binding of other element at the promoter. Next one is TF2E. This TF2E helps to recruit TF2H to the core promoter, and it is required for the promoter melting. TF2F has a major function to bring RNA polymerase at the core promoter. This TF2F consisting of 3 subunits, it binds to RNA polymerase 2 and recruit the enzyme at the promoter to form pre-initiation complex. It also recruits EF2E and EF2H at the core promoter. And the last one is TF2H. This protein consisting of 10 subunits. It is having kinase as well as helicase activity. This is the main factor which is involved into the unwinding of DNA molecule throughout the transcription process. It is also involved into the DNA repair mechanism. This TF2H play role into the unwinding of DNA as well as it is having the kinase activity which play a significant role to carry out the phosphorylation of RNA polymerase CTD tail. Now how it will involve, that we will discuss in detail during the elongation process. But if we understand sequential binding of component, first of all TF2D comes and binds to data box, then after TF2B will bind, then after TF2A will bind. After this TF2F along with RNA polymerase comes and binds to core promoter, and at the last TF2E and TF2H will bind. And this is the way how sequentially all the factors bind along with RNA polymerase to bind and form pre-initiation complex. You can see over here TF2D, TF2A, 2B polymerase, 2F, 2E and 2H. This is the order of binding of different components at the transcription core promoter. This picture again you can see how the protein factors bind and form the pre-initiation complex. After assembly of RNA polymerase and the transcriptional factors, some conformational changes takes place in both DNA as well as polymerases. And these changes cause 10 to 15 base pair of DNA surrounding the transcriptional start site to separate. The single-stranded DNA serve as a template and synthesis of RNA molecule takes place. This single-stranded DNA take its position at the active site of RNA polymerase, and the resulting structure it is known as open complex. Once the open complex forms, the synthesis of RNA begins. And this is the way how the process of transcription is initiated. The last phase of the transcriptional initiation is promoter clearance. After the first bond is synthesized, the RNA polymerase must clear the promoter. Means it should start to move towards the downstream position. And during this time, there is a tendency that RNA transcript get released and the process of the transcription is freeze, which is also called as a abortive initiation. But this can be prevented by the role of different factors and phosphorylation activity that we will see into the elongation process. But once the transcript reaches approximately 23 to 25 nucleotides, it is no longer sleep and elongation can continue. This is an ATP dependent process. So this is the way how the process of transcription is initiated in eukaryotic organisms. The mechanism of elongation we will discuss in our next lecture. Thank you very much.